Dear students, now I am going to start Animal Biotechnology in paper 6.2. In that, genetic engineering, today I am going to discuss the scope and basic concept of genetic engineering. Genetic engineering can be defined as techniques of manipulation of gene or DNA to get desired effect or product. Genetic engineering involves gene cloning or recombinant DNA technology. Gene cloning is nothing but it is inserting desired gene or DNA sequence into a vector and made to replicate and produce numerous copies gene in easily multiplying host cell or organism. So it is gene cloning, you know the clones. So it is a method, the vector will be inserted with the desired gene for which we wanted many copies. So then that is will be inserted into the fast multiplying organism, like bacteria, so that they make multiple copies of our gene of interest. We can harvest gene of interest later, or else the proteins produced from them. So recombinant DNA technology techniques of isolating gene of interest, inserting gene of interest into the vector, then gene cloning, producing many copies of that, and then transfer this to the host cell to get a desired effect or the product. So genetic engineering allows to alter DNA directly. It is the process of transferring the DNA from one organism to the another organism. Then it is recently, so it is helped to develop very fast the vaccines for COVID-19. So this recombinant DNA technology, the way in which genetic material from one organism is artificially introduced into the genome of another organism and then replicated and expressed by the other organism, was contributed mainly by the work of Herbert Boyer, then Stanley Cohen and Paul Berg. So they are responsible for developing this technology. So many other scientists contributed, mainly these three scientists contributed this recombinant DNA technology, that is the basis of this genetic engineering. What is recombinant DNA technology? The ability to combine DNA to one organism with the DNA of the another organism. So here you can see the how exactly the DNA can be a short fragment of DNA can be removed and it can be inserted into the genome or the DNA of the other organisms with the help of the ligase enzyme. So that with the help of restriction endonucleases, it can be removed from the host organism. So recombinant DNA technology was first used in 1970s with bacteria. So it is developed in way back in 1970s itself and created recombinant bacteria. These the pioneers in that the genetic engineering. So they created the bacteria, recombinant bacteria. So what is the basic concept of genetic engineering? What are the main aspects of genetic engineering? So first thing is isolation of gene of interest. So we want to remove, for example, we want to produce large amount of insulin, then we have to first isolate the insulin gene. So that is with the help of restriction endonucleases enzyme. It is a molecular scissor. It acts like a molecular scissor. With the help of this, we can able to cut the DNA sequence of interest in a specific way. So that is what you can see, the molecular scissor. Then we can able to isolate that gene from the rest of the chromosome. So we will use this enzyme and we can able to take out the desired gene from the chromosome by using suitable enzyme restriction endonuclease. And after that you can see See, this is how exactly the example is, we can remove insulin gene or else interferon gene, like that you can see. Then gene cloning, gene transfer to the host cell. So this is what you can see, we have to we need a vector or a molecular vehicle for that. So this molecular vehicle will be always a small circular DNA. So this we have to cut in a specific way, and we can, whatever the gene of interest, we can insert, and we can make this organism to multiply this. So this is what you can see, molecular cloning or gene cloning. So restriction endonucleases are called molecular scissors. They cut specific sequence of DNA. So scientists use this restriction enzyme to cut and paste the DNA. So that is the starting point. Then it requires a vector. So a molecular vehicle to carry the DNA fragment of interest into the host cell for gene multiplication. So that is what you can see. So then using restriction endonucleases, vector DNA cleaved incubated with the desired gene along with DNA ligase. DNA ligase, you know that molecular stitch, annealing and ligation takes place. Desired gene get inserted to the vector. So the DNA resulting now is called recombinant vector DNA. So this is the steps you will able to see. So this is the cell, we have the gene of interest here. So we have removed that gene, and this is the vector from the bacterium. So we have inserted this gene into this vector. Then we have transferred this, you can see the vector along carrying inserted gene into the bacterium. Now it is called recombinant bacterium. This is called recombinant vector. Now the recombinant vector carrying bacterium is called recombinant bacteria. So with this recombinant bacteria in a test tube, we can able to culture in a large vessels or a test tube. So within no time, within an hour, you will saw thousands or millions bacteria will be produced. So each time there is a DNA replication. So this inserted gene also replicated. This is how easily we can able to produce multiple copies of desired gene. Or else we can also you can harvest the protein produced by the desired gene. So that is how the genetic engineering works. So there is after the vector DNA, this is the vector DNA, it has to be transpected, transferring this recombinant vector into the host cell. Then this bacteria, so it replicate in large number quickly and produce desired gene product in large amount within no time. So that is the advantage of this So what are the scope of genetic engineering? So it is now, it's a it is the one, it is there is we can't able to discuss within 5 minutes. So it is such a huge benefits we are getting, and also lot of scope in this technology. So there are so many DNA tools generated out of this technology. So that is for restriction endonucleases, molecular scissors. Then you have studied electrophoresis, how we can able to separate the DNA and protein depending on their size. Then oligonucleotide technology, polymerase chain reaction. So if the DNA fragment is smaller, below 5000 base pair, then we can able to get multiple copies, millions of copies of this within no time using this polymerase chain reaction. We can develop a DNA probe, and we can able to identify these desired gene, or else so hybridization technique, sequencing technique, all this we will discuss later in detail about this. That's how the different DNA tools developed from this technique, which are useful in so many agriculture, industry, medicine, research and all, Similarly, so many technology developed, gene or DNA isolation technology, splicing technology, cloning vectors, development of engineered cloning vectors. So then development of cloning host, engineered host, then the isolation of the gene libraries. So all these technology developed because of this genetic engineering. Then recombinant products, so many products now in the market, hormones, drugs, enzymes, pesticides, probes, vaccines, all these genetically engineered products. So similarly, you will find transgenic organisms. So many microbes are transgenic microbes developed. So they are useful in frost-free bacteria, bacteria with the insecticide, viral vaccines, then nitrogen fixing bacteria, plants, agrobacterium. So then pesticide resistant plants, bt cotton, that disease resistant the plants, so many in the field of agriculture. Then in the field of live animal husbandry, so recombinant embryos, animal models of diseases, mammals engineered. So all these because of this genetic engineering, so many it's a vast area, so we can't able to discuss everything here now. So it is up to you to advance your knowledge with respect to all this. Then genetic analysis, we can map the gene. So there is a human genome project, and we have analyzed the genetic text of the human, likewise Drosophila, so many rice and all the other plants, those are useful. Then sequencing junk DNA, we have identified what is the importance of junk DNA or else non-coding DNA. Then genetic screening for diseases, hereditary diseases, DNA fingerprinting, the importance of DNA fingerprinting, you are going to study also. Then genetic treatment, gene therapy, then so many, these are all the scope of genetic engineering. We will discuss all these. The molecular tools, I am going very fast because it is a lot of tools developed recently. So that is polymerase chain reaction, restriction enzymes, they are called molecular scissors. Gel electrophoresis to separate DNA fragments. DNA ligases are they are developed, they are called molecular stitches. So they can able to make a covalent bond between nucleotide chains. Cloning vectors, molecular vehicles. So many cloning vectors developed to transport the big gene or else the so they can be inserted into the microorganisms, and they can be replicated it into large numbers. Transformation, transductions, all these you can see the technologies developed to clone the gene of interest. Then identifying the transgenic organisms, so whether the our desired gene is expressed by the organisms or the bacteria or the plant. So there are technology developed to various tools and technology developed to identify this, the southern blotting, northern blotting and all this you can see. Then genomic libraries, so restriction endonucleases are useful in creating genomic libraries. So we no need to isolate a gene of interest, we can just approach genomic library, they will provide the gene of interest, they have that. So this is how the technology improved, cDNA library. So this is what you can see, similar genomic libraries you can see. So then CRISPR, recent one you can see, the Nobel awarded technology CRISPR, clusters of regulatory interspaced short palindromic repeats. So this is a gene editing technology. The genomes of living organisms may be modified. It is based on bacterial CRISPR-Cas9 antiviral defense system. So this is what you can see, the recently it is through this technology, so we can edit gene. It is a boon for so many plant diseases and animal diseases like cancer. So genetic engineering has applications in medicine, research, in industry, agriculture, so wide range you will see. So some of this it's not complete, so just I am showing you a tip of the iceberg, that that can I say. In the field of medicine, genetic engineering in the life-saving drugs such as you can see interferon, the development of the insulin, you know, the humulin insulin, then interferon, clotting factor, growth hormones, human albumins, monoclonal antibodies, tumor necrosis factor, erythropoietin, antioxidant enzymes, serum albumin, relaxin, so many, you know. Then vaccine development, you know, the how fast we can able to develop vaccine recently for the COVID-19. So many genetic engineered vaccines, Novavax and Pfizer vaccines, then all these are genetically engineered vaccines, you can see. So within a year we could able to develop because of this advance in the genetic engineering. Just imagine the polio virus. Recently we eradicated polio from from this globe. It took some 50 years to develop that vaccine. The advancement in the genetic engineering, within a year we can able to develop this COVID-19 vaccines. So similarly, half simplex vaccines, vaccines for polio, measles, rabies, so many, you know. So then disease diagnosis, you know, DNA probes, blotting techniques, DNA profiling, all these, you know. So they made very fast, accurate diagnosis of viral. You know how exactly RT-PCR now to diagnose the presence of the coronavirus and you know the so many bacterial diseases, HIV, STD, all this you can see, tracking congenital defects. So there are more than 500 genetic diseases in human: cystic fibrosis, sickle cell anemia, hemophilia. So DNA probes developed blotting techniques, DNA profiling techniques, DNA fingerprinting profiling means all these are helpful in detection of these genetic defects at a fetal stage, and proper therapeutics and measures will be taken to tackle these defects. So similarly, gene therapy, it is a fast developing therapy, involves the replacement and modifications of faulty genes to restore and enhance the cellular function, to improve the response to non-genetic therapies. So all these you can see, to it will be a very, you know, for the cancer, cystic fibrosis, heart diseases, diabetes, and all this gene therapy will be the you know the therapy of the one developing therapy, and some experimental you know the results also we have. Antibiotics, genetically engineered microbes are very helpful in extraction of so many antibiotics, erythromycin, neomycin, carbamazepine, so many. So then in the field of agriculture, genetic engineering to create genetically modified crops, genetically modified organisms to improve the quality and to improve to remove the unwanted traits, insect resistant apple, long lasting tomatoes, potatoes. Genetic engineering techniques, plant breeding, can modify the genetic makeup and creating improved plant varieties which are faster and easier than traditional plant breeding techniques. Golden rice, so it is carrying the you know the vitamin A, beta carotene. So then BT cotton, BT brinjal, see that this is the normal brinjal, so many, you know, the insects going to you know spoil this. This is the BT brinjal. Then you will find the advances made in developing crops that mature faster and tolerate aluminum, boron, salt, drought, frost, other environmental stressors, allowing plants to grow in condition where they might not otherwise flourish. So the genetically modified, you know, the soya beans, so much of yield, you know. Then you can express the you know the omega fatty acids, or else important DHA, or else decohexa. This is the fish oil we can express in a canola oil. So canola, so this is what you can see. We can increase the genetic diversity, production of variant alleles, possible to alter genetics of various plants and animals and microbes. So through this, you know, this is what you can see, genetically modified. So how exactly? Not you know, this is the engineered one. So you cannot it. So similarly, the increased pulp content. Similar, you will find the quality in the maize. So BT brinjal. And see the this is what you can see, the in the pest resistant, the BT cotton. So this is the normal cotton. See the yield because of this worm, ball worm. So this you will find it is eating the leaves, and photosynthesis is get reduced. Here BT cotton, see it is almost 10 times more yield you will able to see. So similarly, improvement in the livestock, you can see gene manipulation in animal husbandry. So the growth hormones, and so that the dairy animals, poultry, see this, then fishery, the salmon, the genetically engineered in the normal, it is 10 times improvement in the amount of the muscles, you can see. So the transgenic livestock, so that is the improve the human health, enhance nutrition, protect the environment, increase the animal welfare. So many advantage one can think about. You can see the improve the livestock, stem cells, and other emerging biotechnologies will have important for producing, because it is the increasing population, that is the only answer what we have. So the transgenic animals, you can see the mice extensively used to do research. Then in the livestock, in the chickens, you can see resistant to infections, you can see. Then increased milk supply and leaner meat in the case of cows. Then human proteins can be harvested in the goats and the pigs. So those are all the. Then main issues are environmental friendly technology, bioremediation. So we can able to take care of oil spills, sewage treatment, industrial waste. So all these are with the help of this engineered microbes, you can see. Biomining, we can able to enhance the ore extraction by this method. Then we can reduce the carbon in the environment by sequester the more carbon dioxide from the environment through the engineered the plants. So many genetically engineered organisms can help us to diminish atmospheric carbon and tackle climate change. So on this the poster will represent how we can able to tackle this problem. Then there are so many xenoplant transplantation, genetic engineering in the research, so many technologies are developed. Then genetic engineering in the industries, varieties of industrial products, detergents, lens solutions, recombinant microbes used in making the so many polyesters, polymers, and all this, you can see. So that is the only answer for us now left when it is from 2006 to 2050 we will have a gap of 70% food, that food gap we have to fill up only through this genetic engineering technologies. So genetically modified foods are really just better crops. They are more nutritious, they are more resistant to drought or pests and other sort of things. So this is what the importance of genetic engineering. Thank you.